In this webinar, I'll explain the principles of the myeloid cell therapy and how it could be used in settings of cancer. In slide 2, we see the underlying hypothesis for our clinical treatment paradigm. This will be that one will isolate a specific cell type, monocytes, from the blood of an individual. One will manipulate them, specifically activate them through in vitro culture, and then adoptively transfer them back into the same individual in order to effect therapy. In the next slide, we see a cartoon to try and explain the basis of why this therapy will work. On the right-hand side, we see the balance of immune reactions in a resistant, healthy individual. In the upper panel, you see a yin-yang diagram showing that there's balance between pro-inflammatory in black and anti-inflammatory in white activities. This doesn't mean to say that this individual cannot mount an immune response, but it does mean that once this response has been mounted, then it will be down-regulated. On the left, we see what happens with an individual who will have a disease state. There will be an imbalance in the yin-yang, either in this case more pro-inflammatory, and this will be typical of an autoimmune individual, or in the case of cancer, the portion of white will far exceed the portion of black, giving a higher anti-inflammatory state to the individual. So the idea of the myeloid cell therapy is to restore the balance and the situation that you see on the right-hand side. In the fourth slide, you see an animation of how efficient tumor killing should occur in a healthy individual. The tumor mass is the orange cloud on the right, should be efficiently disposed of by the combined action of cytotoxic CD8 lymphocytes and natural killer cells. In this next slide, we'll see an animation trying to depict how the tumor defends itself. The tumor mass is associated with other cells with immunosuppressive activity, regulatory T cells, tumor-associated macrophages, and myeloid-derived suppressor cells. This combined activity of these cells will enable a sort of force field around the tumor to be immunosuppressive activity. So even if the immune system mounts an anti-tumor response with its CD8 and NK cells, these will be neutralized when they come into the vicinity of the tumor mass. So this blue zone of immunosuppressive activity is protecting the tumor and neutralizing the activity of anti-tumor immune cells. One anti-tumor therapy which is tested today is vaccination with dendritic cells. These are other immune cells which are loaded with tumor antigen, and the idea is then one will expand their numbers. This has been tried in the clinic with very poor clinical efficacy, and we would suggest the reason would be is that even though one increases the number of these anti-tumor cells, the immunosuppressive activity around the tumor is still dominating, and so the induced immune cells are inhibited in much the same way as we saw on the previous slide. In this last slide, we see an animation of how we expect our myeloid therapy to work. In order to combat this immunosuppressive force field, we would inject a high number of pre-activated pro-inflammatory macrophages, here in red, at the site of the tumor. Being in excess of the immunosuppressive activity, the pro-inflammatory macrophages will then immunomodulate this region, reducing the immunosuppressive activity and then allowing for efficient tumor killing in the settings, for example, of dendritic cell vaccination. The important point here is that without addressing the local tumor environment and an immunomodulation, the dendritic cell vaccination will not be efficient. But in this case, if you do the immunomodulation on site, then it will be. This is how we envisage one will be able to treat many different types of cancer, and you can follow our work on this homepage.